So this video is going to focus on some of the HL additional topics that focus on amino acids. So the SL class should know that amino acids can act as buffers, but in the HL portion of the topic, you're actually going to do some calculations with buffers. Now, this used to be part of the HL core anyways, so it's not such a big deal to have to add it in now. But let's get looking at the amino acids. So as I mentioned in the SL video, amino acids can act as buffers because they contain both a weak acid and a weak base. And so if you can think back to our acid-base topic, that's essentially what any buffer is. It's a weak acid and its conjugate, but in the amino acids case, it's because it has those two different functional groups. So if you think about a Zwitter ion, it contains both this positively charged H+, and it contains this negative OH-. So we're going to be able to accept protons here, and we're going to be able to donate protons here. So buffers are amphiprotic, and that's what makes them such good buffers. So they tend to buffer around particular pH values. Now, the calculations for the pH of any buffer, so a little bit random, but Ivy's going to perhaps ask you some buffer calculation questions right here, is based on some of those ideas that we've done before. First off, if you think about the concentration of any weak acid or any weak base, we're going to assume that its concentration at equilibrium is equal to its initial concentration. So you're always going to be allowed to assume that when you have a weak acid or a weak base, that their concentration is whatever you're told in the question. The assumption for the concentrations of the conjugates is a little bit trickier. It depends on which method you're using to make your buffer. If you're using the method 1 where you add salt, then you're going to get to assume that the moles of the salt equals the moles of the conjugate, because the salts tend to be quite soluble, so they completely dissociate into ions. So however much salt you add, that's how many moles of the conjugate you're adding. If you choose to do it the other way by adding the opposite strong thing, so if you're starting with a weak acid, if you add a strong base, then the moles of the strong thing are going to be equal to the moles of the conjugate, because the strong thing will completely neutralize a certain portion of the weak thing that it's reacting with, and that will all turn into conjugate. So the way to figure out the moles of the conjugate, it's either equal to the moles of salt that you're adding or the moles of the strong thing that you're adding. Now, you can just use the original Ka expressions like we always have, and you can solve for pH that way, but you can also use a reworked version of the Ka expression known as the Henderson-Hasselbalch equation. This is in the data booklet under B7 and D4, so it's a little bit later on in the data booklet. And I can show you in class how this is the exact same as the Ka expression. It's just a little bit more convenient for buffers. So if you're doing a buffer calculation, you can look up the pKa value in the data booklet. And so all that you need to know to calculate the pH of a buffer is you need to know the moles of the weak acid and the moles of the conjugate base. This would also work for a buffer made from a base, except this would be the pKb, these would be the base and the conjugate acid, and this would be the pOH. So with a base, you just remember it's always pKb's and pOH's. But these two things I just went over up above, how you can find them, and really you're just plugging in. And you can see right here, it's even clearer than with the Ka expression. If these two concentrations are equal, so if the weak acid and its conjugate are in exact equal amounts, then this becomes a 1, the log of 1 is 0, and pH equals pKa. So when these two things are 50-50, the pH of a buffer will be equal to the pKa of the weak thing, the weak acid it was formed from. Now, actually, each amino acid has 2 pKa values, and I'm not sure if this would really be touched on the IB exam, but the InThinking website had some questions on this, so I thought I'd involve it. You can think about it that this, actually first, this proton can get donated at very low pHs, and then this is the second proton that can get donated. So there's actually it sort of acts like a diprotic acid in that way. And so each amino acid actually has two pKa values. There's a pKa value for this first acid, and there's a pKa value for this second proton, the second acid that can be dissociated. And so as the previous buffer content has gone over, at the pK, when you have species in 50-50 concentrations, then the pKa equals the pH. So for example, alanine has two pKa values. They're 2.3 and 9.7. So at a pH of 2.3, what do we know about alanine? Well, the pKa of 2.3 is referring to this first acid. And what that means is at 2.3, you'd have 50% this positive form, and you would have 50% Zwitter-ion form. So it's actually a little bit more complex than the SL questions where it just says, which form is it in? It's going to be a mixture of forms, but you're drawing the most dominant form. At the 9.7 being the second pKa value, so that's this proton, what it's referring to, how strong that acid is. And so at pH 9.7, you'd expect to have a 50-50 mixture of the Zwitter-ion and the negative form of the amino acid. So you've got those two pKa values, refer to those like two values that the amino acid could buffer around when you have 50-50 of these two forms and then 50-50 of those two forms. The last topic to go over with amino acids, this HL, is that every amino acid, with the exception of glycine, because glycine's R group is an H, every single amino acid besides glycine is chiral or is optically active. Because this carbon here is the chiral carbon. It has 4 distinct groups attached to it in all examples except glycine. So you should be able to draw enantiomers. So the dash wedge structures where you show how they are mirror images, you draw the dotted lines down the middle, and you use dash wedge to show how they are mirror images of each other. So I'd like you to try to do that as part of your homework. If you're looking at this video, go look up alanine in the data booklet. The R group is a methyl group, CH3, and attempt to draw the 2 enantiomers. Now, what's really cool about biology and biochemistry, amino acids in general, related to this topic, is that while both isomers are possible, both can be made in the lab, in nature, only one type of the amino acid is ever found in humans, and that's what they call the L form. The one way to talk about optimal isomers is L isomers and D isomers. That refers to the direction that the light gets rotated. But it's pretty incredible that our body only produces one of the optical isomers, one hand of the amino acids. Whereas if you made this in the lab, you would make 50-50 of each, and it would be really hard to only make one side. And that's important because your body is incredibly stereo-specific, and having the right hand, right? Well, your left hand only fits into your left glove. And the same thing goes for amino acids, that only the left-handed version is going to bind up properly with certain substrates. If it's in an enzyme, for example, the body is incredibly specific. The reactions that it undergoes are very stereo-specific, and so this L kind is going to be found in our bodies. The D kinds are pretty rare in nature, but they can be made, and they are used in certain drugs and things like that. But for the most part, only one type of amino acid shows up in our bodies. So here's some practice questions, mainly focusing in on the buffers. And we've covered these additional topics on amino acids now, about them acting as buffers, doing buffer calculations with the Henderson-Hasselbalch equation, and talking about the chirality of amino acids.